Today's topic is cytology, the study of cells. So we're going to jump right into this and see what we can discover about cells and cell function. The human body has over 200 different types of cells, and each of them have their very specialized functions. So we are going to look inside of the cells and see what the special organelles that are found in cells, what their special functions are, and how they function. This is incredibly important because we understand that all disease is actually at a cellular level. How these molecules come together to form these macromolecules that form the organelles that compose our cells. This is where the problems happen. This is where things go wrong. So this chapter is dealing with cytology, and it is the study of cells, and I just can't emphasize more the importance of this. As we go through this, a great way to be studying this is if you actually, when you hear about a particular structure found within a cell or on the outside of a cell, that you listen closely to what its special function is. And if you could match functions to these structures, then you're going to be okay. But please realize that the mechanisms of disease, all of them are happening at this molecular and cellular level. So to be able to truly understand how to treat diseases, there has to be a very much of an understanding of how these cells function. So cytology, again, is the study of cells, and we understand that it was really Louis Pasteur that actually, with his experiment around 1900, he was the one who finally, after about 400 years and dozens of scientists, his experiment finally let us accept the cell theory, which is a scientific theory that we said is the most exalted explanation of how something's happening in our natural world. And so the theory of spontaneous generation, which had been thought to be how life came about, where life just sprung up from non-living things, had to be put to rest and had to be let go of. It was a really difficult thing to do, to let go of the theory of spontaneous generation. As I said, it took hundreds of years of researchers and scientists actually using the scientific method and doing innumerable numbers of studies before finally that was let go. And the reason for that is because people really couldn't see at a microscopic level, and also you only know what you know, and it's only when you start to actually accept evidence that's before you and you have enough numbers that support that evidence that you can actually integrate that information and accept new information when it comes in. So that's really what science is all about too. So I do want you to know that Louis Pasteur, we sort of give him credit for the cell theory. And the cell theory simply stating that all life are composed of cells, and cells are the smallest unit that is alive. And then also that all life comes from pre-existing cellular life. So all life comes from pre-existing cellular life, and a cell is the smallest thing that we know that is alive. Now we're going to go inside of cells, and we're going to look at these organelles that are made of these huge molecules. But organelles are not alive. Molecules are not alive. The fluid that's inside of the cell is not alive. But all together, when the cell has just what it needs, there is this amazing thing that we call life that is happening, where all cellular life as we know it can extract what it needs from the environment, change those molecules into usable sources of energy so that the cell can actually do work. Cells can never completely rest. They're always having to sense the environment, adjust to the environment, make changes because of the environment is constantly changing, and then to be able to do their work. And so it is a 24/7 thing. If anything gets disturbed to the cell, then the cell could die. So we understand that not all cells are equal in a human body. We have, again, more than 200 different types of cells that have different shapes, structures, have different sizes, and they have different lifespans even. Some cells that we make, our bodies are creating, will only live for a few hours, and other cells will live for days. Different types of cells might be required to live for decades. So it depends on what information the cell got, what the cell differentiated into and matured into, as to how long the cell will live, what size it will be, what shape it will be, and what product it will produce. So as a reminder, let's make sure we understand that sexually reproducing organisms, which Homo sapiens are, we start off as one cell. That one cell is called a zygote, Z-Y-G-O-T-E. And as that cell starts to undergo cellular division, which is called mitosis, that one cell will make a copy of its DNA. The two new cells that will form will each have an identical copy that the first zygote had, and one cell becomes two. The two cells will undergo the same type of cell division, mitosis, and two cells become four, and on and on and on. The first cell's a zygote. That's when an egg and a sperm meet and fertilization occurs. And the zygote, until about 2 weeks of development, is called a blastocyst. Blast, B-L-A-S-T-O, cyst, C-Y-S-T, one word, blastocyst. The first 2 weeks of dividing, this newly dividing group of cells is called a blastocyst. From 3 weeks in the uterus until 8 weeks, that newly dividing organism is called an embryo. From the 9th week until birth, it's known as a fetus. And from birth until 6 weeks, this is referred to as a neonate. So it's important that you understand that progression. And we understand that even though as an organism, a neonate has trillions of cells, we understand that it started as one cell. So that one cell that had that DNA, that recipe book that had never been written before, it all starts with the zygote. Now, after trillions of cells are created, the cells have unique functions. They're going to mature, or what we call differentiate, into a very specialized cell. Those differentiated, specialized cells will have their own unique shapes. They will have their own unique sizes. And depending on what part of the DNA opens up for them to act the way they're supposed to act, that's what we mean by maturing or differentiating. Your liver cells that are unique, that produce particular enzymes, they're not going to all of a sudden be able to change into skin cells that are producing keratin. Your skin cells that produce keratin, they can't all of a sudden change into other types of skin cells called melanocytes that produce melanin. They go along a certain pathway, they differentiate, they mature, and once they become what they mature into, they can't change back. So we talk about cells maturing, but I now want to give you the concept of an early cell, one that hasn't chosen its pathway, one that has the potential to become any type of cell: cardiac muscle, skin, nerve cells. It has the potential to become anything. These early cells are called stem cells. So a stem cell is an early, undifferentiated cell that has the potential to become anything that the body may need. So I hope that kind of clears up some of that. But let's get back to looking at these different cell types. There's some terminology that's used when talking about the shapes. The shape, squamous means kind of amorphous, meaning there is no morphology. A as a prefix means without, right? Morph means shape. So without morphology. Squamous cells, they're kind of influenced by their environment, so they're kind of flat and squishy, and they really have no real shape. Polygonal means that they have multiple sides and shapes. Stellate is star-like. Cuboidal, as it suggests, is like a cube. Columnar is like a column, tall and sort of skinny. Spheroid is like an orb, if you will. Discoid is like a disc. Fusiform is sort of fat in the middle and pointy on the ends is what that means, and fibrous is thread-like. So there are many different shapes, there's many different sizes, and as I said, depending on what the cell differentiates into, there's different life cycles. As I said, some will live for minutes, some hours, some, well, hours really, hours, some days, and some will have to live, will have to be there for decades. So it just depends on what they differentiated into. These are some illustrations that are showing you the different types of cells and their shapes, and you should be familiar with those terms. So as I said, the sizes differ. Now, when we are looking at cells under a microscope, because they do require a microscope to be able to visualize them and get any detail, usually a light compound microscope is really the only thing that's needed in medical cytology for human anatomy. And we can see particular structures in those under a light compound microscope. But to be able to see inside of cells and be able to see the structures that are made, are making the cell perform the way that it does, it takes a higher magnification of a microscope. And so we often use electron microscopes. They're the ones that have given us the information about what these structures inside of cells look like. And that anatomy, the structure of those organelles, have helped us to understand what the functions are. So to really be able to see inside of a cell and what's going on, it takes a much more powerful microscope than just a regular light compound microscope. When we talk about resolution, it just simply means that between two points, you can actually distinguish between 2 points. So in a light compound microscope, it's a little bit fuzzy here, but looking at the electron microscope, you have much better detail. So I think that's all for right now. I'll have a lab that I'll talk about the microscope, so you all will be able to learn more about microscopy in that particular lab. So when we think about the basic components of a cell, there's some things that we learn about that once we learn about their functions, they have the same function no matter what cell you're talking about, no matter what the cell matured into or differentiated into. That organelle, that structure, it has the same function no matter what the cell type is. So this is what I meant about when we are talking about a cell and we are looking at the different structures inside of a cell. And here's one cell that's been cut open so you can see it has a neighboring cell here and a neighboring cell here, but they've illustrated some of the structures inside of this cell. This is what I meant about listing them and making sure you could match their functions to their structures. The cytoplasm, plasm means fluid, cyte means cell, so the fluid part of the cell, cells are filled with fluid. Of course, there are particular structures inside of that, but cells are filled with fluid, and that fluid is known as the cytosol or cytoplasm. And inside of the cells that are three-dimensional, you know, if cells were truly flat the way that they look on pictures, then we would be flat, but they are three-dimensional. This is where we're going to find inside the cell, inside the cytoplasm, inside this fluid matrix, we're going to find these organelles. We're going to find what is called the cytoskeleton, which I call the highway of the cell. So where things get shipped. So as something might be made here, it's going to be shipped here and then eventually shipped out. So the cytoskeleton we'll talk about too. And again, the cytosol is actually fluid, the fluid part of the inside of the cell, intracellular fluid. There's also fluid bathing all of our cells. So the fluid that is bathing our cells is called extracellular fluid. And we'll also talk a lot about extracellular fluid, so you'll know that I'm talking about that fluid that is bathing our cells. When you hear about the apical surface, it just simply means the top surface of the cell. So that's what that's referring to. Again, lots of different types of illustrations. So you could maybe say this is cuboidal or columnar. I don't know the aspect here. But then here, this one's sort of really kind of without shape, isn't it? And they've cut a wedge into this cell so that you can, here's the outer, it's all translucent, you can almost see through it, but then they cut a wedge so that you can actually see some of these organelles, these structures inside of the cell. So there's lots of illustrations to look at. I would really recommend people to do that as they're thinking about the cells, but we're going to jump right in here and go ahead and start talking about each of these structures. When they have shown this illustration and they're showing some structures found on the outside of the cell, it's unlikely, it's not, no cells really have all of these membrane structures, but they're just giving you an idea of what they would look like on the outside. For example, these are cilia. These are called cilia, where my mouse is, and these would be called microvilli. There isn't really a cell that would have both of these, but they're just showing you what they would look like if they were there. The first thing, and it makes sense to maybe start with this, is the barrier of the cell. What keeps the cell's structure? What is it that makes a cell have the shape that it has, and what's keeping the inside in and the outside out? And it is the plasma membrane. It's green here for you so that you could see. The plasma membrane is also sometimes referred to as the cellular membrane or the cell membrane. And this is a structure that keeps the integrity of the cell intact. And it's very important that you know what it's made of and how it's doing that. The plasma membrane is said to be selectively permeable, which just simply means that it allows some things in and out and other things not in and out. It's selective in what it's going to allow to permeate past the membrane. So that's all that actually means. Now, how does it do that? How does it do that? Well, we would want to look at the anatomy, because the structure helps us to understand the function. And when we look at the anatomy of the plasma membrane, here we're seeing the surface of it. When we look at the anatomy of the plasma membrane, what we realize is that it's really two layers. When we look closely at it, it is a bilayer of what is called phospholipids, two layers of phospholipids. I would want you, if you would, to look at just this one little phospholipid here. It's got a little head, this is called the head, and it's got two little tails, right? So here's one, and this is lined up, all with other heads here, and their little tails are pointing up. On the outside of the cell, there's another layer, and these heads are all together, and their little tails are pointing in. So if you look, it's like this, right? And the reason is that this molecule called a phospholipid has a hydrophilic head, meaning it likes water, and hydrophobic tails, meaning these little tails don't like water. They move away from water. And it is that property of those molecules that allow this arrangement of these phospholipids, these hydrophilic heads on the outside of the cell, because the cell is bathed in water and it likes water. And then the inside of the cell, which is also filled with water, these hydrophilic heads are oriented that way. And this bilayer is 90% of your cell membranes. And you all might be thinking to yourself, that doesn't seem very strong, but it's strong enough. It is definitely strong enough. It is keeping the integrity of the outside out and the inside in. And most of your cell membranes, your plasma membranes, are lipids. They are lipids, phospholipids, that form this bilayer. And then other lipids as well. Now, how do things actually really truly get in and out selectively? Like our cells actually dictate what it allows in and what it doesn't allow in, are through the membrane proteins. These proteins are embedded in throughout the membrane. And it is these proteins that act as channels, tunnels, receptors. They have all kinds of functions, these proteins do, that help the membrane carry out its functions. But the predominant component of cell membranes, and you see the cell membrane, is a bilayer of phospholipids, or mostly fats. Once you make a cell, I don't want you all to ever think that, okay, well, you've made your liver cells, you've made your kidney cells, you've made your skin cells, so I'm kind of done with that part. I probably don't need as many lipids, as many fats as usual. Wrong. Cells that are alive and are working and are carrying out functions are continually changing out and replacing parts of their membrane. They're continually placing these tunnels, these receptors, these markers, these channels, these proteins. They're continually changing. So this is why, on a daily basis, you should have enough lipids. This is just one example for plasma membranes. You need them for a lot of other things as well. Lipids, proteins, and even carbohydrates. These little rings here, these carbohydrates that will have very specific functions depending on their shape on the outside of a plasma membrane. So, again, what you want to know is that the plasma membrane, the integrity of the plasma membrane, is kept in place by this bilayer of phospholipids. And embedded in those phospholipids are very specialized proteins. On the outside, there are going to be very specialized carbohydrates and more proteins. And it's just important that you understand that. There are going to be cholesterol molecules that are spread throughout. Cholesterols are also lipids that are spread throughout that act like the studs inside the walls of your house. They help keep your house in the shape it's in, right? And the walls standing up instead of collapsing down. So all of these structures are needed. All of them are necessary for the cell's integrity to be maintained. If the membrane is interfered with enough, the cell dies. The cell lyses, L-Y-S-E, lyse means to split. And if a cell splits, it's going to die. It's dying, or already dead. So that's some terminology for you. Okay, so I had talked about a lot and remember, what I talk about is what you need to know, and how I talk about it. Don't memorize textbooks definition, because when you do, you typically you won't recall it, and you will lose it. So make sure that you could talk about the way that I just did, how plasma membranes actually function. It is the proteins that have the diverse types of functions, though. That phospholipid bilayer is kind of boring, really, in its organic structure, wonderfully boring, but it is the proteins that are embedded throughout that carry out so many different functions. They are carrying out so many different functions, really contributing to that selective permeability, really depending on how the cell is going to be targeted by some things. There's some of your cells that, this is just an example, there's some of your cells that can respond to particular hormones and other cells that can't. Now, how is that? It's because the cells that differentiate mature into the cells that need to be responsive to a particular hormone will build the particular receptors, proteins that it needs, and place them in that membrane. All of your cells, and I talked about enzymes, all of your cells have what are called self-identity markers. All of your cells have these self-identity markers, and somewhere in here, that are going to let your immune cells that are circulating around looking for any cells that are foreign or looking for cells that are causing trouble. But all of your cells have these self-identity markers. You can think about them as little flags that say, hey, this is me. I'm fine. This is me. I'm self. This is the self-identification tag. So don't do anything to me. You have immune cells that are, there's one type called natural killer cells. Those immune cells are circulating around in your bloodstream, and they are just looking for something that doesn't have that self-identity marker. And if they see it, they're going to attack it. This is also why when someone gets a transplant from a donor, they have to take immunosuppressive drugs. Those immunosuppressive drugs are suppressing those natural killer cells so that you don't reject the transplant. So, okay. Now you also have some that act as pumps. Every class is important, but the classes that act as pumps are actually allowing the cells to survive on a second-to-second basis, because there's some things that just cannot stay inside of the cell, and it has to be a constant pumping out of those products. I'll give you an example of that. An example of that is, they don't give it to you here, but I'll give it an example of that. One of it is the sodium-potassium pump. Sodium Na, potassium K pumps. And I'll pretend like this is one. So in the sodium-potassium pumps, you're constantly pumping sodium outside of the cell. And potassium is going to stay inside the cell. Sodium is, and you would want to know this, sodium is your predominant extracellular ion, cation, positively charged ion. And potassium is your predominant intracellular cation. Now, that's going to be important for you to note when we start talking about action potentials of cells, and we'll talk about those with muscles and the nervous system, but action potentials and how they're generated. It's important to note that sodium is the predominant extracellular, and that's normal, extracellular cation, and potassium needs to stay inside the cell, predominantly inside the cell. Of course, there's some outside, but predominantly it's inside of cells, and it's also positively charged. You have other pumps. You have pumps like chloride pumps, and when you think about a chloride pump, it's pumping chloride outside of the cell. You all told me that there's sodium out here, and it means that when this is happening, sodium and chloride being pumped out, it gives a nice saline, sodium chloride, a nice saline layer to our cells. You all have heard about us maybe being a little salty, right? Maybe not in that way, but okay. And it is important that there be a normal saline layer outside of our cells. So those are just some examples of that. and go through. Again, if I go through it, you're not going to really need to know it. Now, I need you to know on the outside of your membrane, and everybody's outside of their cells, there's a layer called the glycocalyx. Glyce means sugar, if you will, is a carbohydrate. Glycocalyx layer. You actually can almost think of this as like your sugar coating. I don't know, maybe that'll be a way you remember it. But when you think about the glycocalyx layer, let's see if I have a good picture of that. I really don't, and that's something. But okay, it's on an electron micrograph. I think I have an electron micrograph soon. Okay, here's an actual. These are illustrations and drawings. This is an actual electron micrograph of a human cell that was sliced, and you can see this fuzzy little coating on the outside. This is the outside of the cell. This is the inside of the cell. This fuzzy little coating is actually this glycocalyx layer. This glycocalyx layer, which is mostly composed of carbohydrates. What I want you to know about that is that this is where the self-identity markers are found. So this is where, when someone is going to get a transplant from a donor, they talk about tissue typing and making sure that it's compatible. Well, what they're looking at are these molecules on the outer cells, and making sure that a certain percentage of them match the recipient. Can they find something that's identical? No, not unless someone has an identical twin that wants to donate. But they try to get a certain percentage of them that are the same. And what we have seen through evidence-based research, which is what science is that if we can get a certain percent being the same, then there's a much better likelihood that the recipient will not reject the donation. So where are those structures, those molecules found, those self-identity markers, and those very specialized molecules? In this glycocalyx. That's the layer it's found in. Now, there's something else on the outside that may be found on certain cells. It's not on all cells, it doesn't need to be. But on some cells, we have these structures that are called microvilli. And they look like this. It's in green now. They look like this. They actually look like the membrane, and it's always on the top surface, the apical surface. It almost looks like the membrane has been pushed out into little projections. And that is really what it looks like. And here again, looking at an actual human cell, an electron micrograph, we can see these projections of the surface of the membrane. And what these do, and I hope the anatomy helps this make perfect sense to you, so it's not memorization, it's actually just intuitive, that when these microvilli extend the membrane, and it extends it incredibly, like the surface area is extended really more than 40 times. It's for rapid absorption. So where you have cells that have microvilli are areas where you need rapid absorption. And again, I hope that makes really good sense to you. So for example, when you think about your small intestines, which are about 22 feet long in length, the opening of it, if you look through the small intestines like a tube, if you straighten the 22 feet out and look through it like a tube, the inner lining of the small intestines, that the opening is called the lumen. Lumen is just the hole, if you will. L-U-M-E-N means the opening, and the tube, the empty space inside of a tube. But the lining of that the cells have microvilli. Why? Because as things are moving through the small intestines, you want every opportunity for the nutrients to come in contact with these membranes, and look how much area you have a chance of nutrients coming in contact with, so that you have a chance of absorbing the nutrients. So again, I hope that makes really good sense to you, that where you need rapid absorption, you're going to have cells that have microvilli lining those structures. Now, the next structure we're going to talk about are cilia. Cilia. Again, there's no cell that has both of these on the surface, but cells that have cilia, which are going to be finger-like projections, if you will. Some people say hair-like, and because they say hair-like projections, that's why they call it cilia. Like your, I don't know if you know, but like your eyelashes and eyebrows are cilia. Anyway, hair-like projections, they're not hair, they're not. They're actually projections from the membrane, the surface of the membrane, and there's motor molecules, like almost like, I'm trying to think of how I would describe this, but there are these motor molecules that are inside of these structures that are going to move the cilia. And when you first look at ciliated cells under a microscope, and sometimes you can actually get them and they're still alive, so the cilia is still moving. It looks like the cilia is kind of moving willy-nilly, but it's not. The cilia are actually moving in a very coordinated fashion. In human cells, like this is an illustration of a human cell, here's another one, another one, another one. Human cells that are ciliated, the cilia are moving substances past the cell. The cilia do not move the cell. They move substances past the cell in a directional way. So for example, your lower respiratory tract, your trachea, that what people call the windpipe, that has a lumen, has an opening, you know, where air is moving through. But lining the trachea are ciliated cells that are moving substances up, like an up escalator. When you are breathing air, and you are breathing air that is not sterile, there are all kinds of things in the air that you're breathing right now. Probably some of your own dead skin cells, dust mites, dust mite feces, pollen. There's all kinds of things in the air. It doesn't have to be sterile. It should be clean enough, but it doesn't have to be sterile. But the things that don't get trapped in your nasal area and do make it to the lower respiratory tract, it's okay, because there's a nice saline layer, thin little mucousy, with mucus, and mucus is okay, that's a good thing. It's trapping the debris, and with that thin saline layer, it's moving it like an up escalator, anything that could get that is trapped in the air, so that really your actual air is getting to the lower lungs and nothing else, hopefully. So that's just an example. Another example of where you find ciliated linings of tubes are the fallopian tubes from the ovaries that connect the uterus to the ovaries. Those fallopian tubes, the inner lining is ciliated, because you all know fertilization doesn't happen in the uterus. It doesn't happen here in the uterus. It happens out near the ovary. So sperm have gone a long way to get out there. They've gone through the cervix, which is the opening of the uterus, through the fallopian tube, and gotten out to the ovary. Fertilization may or may not have occurred. If it did, the zygote, which is now starting to divide, becoming a blastocyst, is got to move all the way back down through the fallopian tube to the uterus. Do you think it just does that through gravity? No, the inner lining of the fallopian tubes are ciliated, so that blastocyst is gently being encouraged to move down through the fallopian tube to the uterus. It takes about 6 days for that to happen after fertilization occurs. If it happens as it's directed, then implantation happens about the 6th day after fertilization. And that's when people say they are pregnant. Okay, so where we need cilia, which is really important, where we need cilia, we need them to functioning by moving substances past the cell. Make sure you know that specialized function. Okay, now here's an example of when things go wrong. So I had talked to you about pumps, and I'd said we have all kinds of specialized pumps, which are really just protein molecules that are carrying out specific functions. None of these things, by the way, are alive individually. They only have this animation doing this work when they're inside of a cell, and the cell is directing this work. Okay? But there is a special kind of pump called chloride pumps I talked to you about, and they need to be pumping chloride out. There is a disease that is known as cystic fibrosis. This is an inherited, whenever I tell you all something is inherited versus autoimmune versus contagious versus communicable, these are important terms because they're very different in how someone has these diseases. If it's inherited, it meant that it's at a DNA level. So it was actually a disorder that was inherited from the parents. Sometimes it takes both parents contributing a trait for a disease to happen. Sometimes it only takes one parent to contribute the trait. In this particular disease, cystic fibrosis, it would take both parents having a recessive trait, that recessive trait came to the offspring. So both parents contributing. It is an inherited disorder of the chloride pumps. So what happens, instead of there being a nice saline layer, mucus is normal and you need it. But instead of it being a nice saline layer moving that thin mucus, there isn't a nice saline layer because the chloride pumps aren't working. So the mucus starts to build up. And because the mucus starts to build up, then the cilia can't move substances past. So in the respiratory system, this is going to end up leading to many respiratory infections, respiratory compromise, and oftentimes cystic fibrosis patients, that's their demise, is because of respiratory issues, chronic respiratory issues. But the cilia that are also found in the digestive system and whatever can't work either. So nutrient depletion and poor nutrition due to malabsorption, can't absorb, wouldn't matter how much they were trying to eat, they can't absorb, is also happening. I want you to note that life expectancy, and there are a couple of different variants, genetic variants of cystic fibrosis, but of the typical cystic fibrosis, that the genetic variant that we typically think about, a 30-year life expectancy is pretty good, and that usually is also going to have required maybe lung transplants. So now with lung transplants that usually last they try for them to last about 15 years. They're happy if that happens, and then they would require another. With those, life expectancy can be extended, but this is a horrific, cystic fibrosis is a horrific inherited disorder. Again, and what is it affecting? It is affecting faulty chloride pumps, and you should know that. Okay, so the next structure that may be found on the outside of a human cell, there's actually only one human cell that has these, are called flagella. Flagellum, which is singular, flagella plural. Flagellum is a tail-like structure that whips, and again it's going to cause the movement of a cell. The only motile cell that has a flagellum in the human is a sperm. So in males it's the sperm. So the movement they're talking about being undulating or snake-like And again, it's because of just the way that those motor proteins are actually functioning. So, plasma membranes. How exciting are plasma membranes? So we have plasma membranes that, due to their bilayer, 2 layers of phospholipids, are keeping the outside out and the inside in. But embedded in them are these special proteins. There's carbohydrates, there are other lipids that have very specialized functions in making that membrane selectively permeable. Some cells have microvilli. Some, depending on how they need to be functioning, how they differentiated, how they matured. Some cells will have cilia that move substances past a cell. And you didn't see a sperm cell, but y'all can look up what a sperm cell looks like. And then some of them actually on the outside would have a flagellum, which we said are the only ones that are going to be sperm. So what we're going to talk about now is a little bit about a couple of terms that are related to how things move in and out of a cell. So I want you to write down 2 terms for me. I want you to write down the term diffusion, just diffusion, just that word diffusion, the word diffusion. And then I want you to write down the word osmosis. Now, these are not related to the same things. When you are talking about physiology, like say cell function, diffusion is actually a term that when we think about diffusion, it is going to be the movement between 2 areas of concentration. So for example, from outside the cell to in, or inside the cell to out. So 2 areas of concentration. The movement of solutes, S-O-L-U-T-E. Please spell that correctly. T-E. S-O-L-U-T-E. The movement of solutes. Now here they're saying particles. Okay, that's fine. Solutes. Now, that's diffusion. Movement of solutes between 2 areas of concentration. Now, the movement of water between 2 areas of concentration, water, is called osmosis. Now you might be asking, what is a solute? What is a solute? And here's the word written for you. What's a solute? Here's one, and here's a little purple one, bigger one, solids. What are solids? Solids are any molecules other than water. That's just a good rule of thumb for you to know. So even gases, like O2, is a solute. Glucose, C6H12O6, a solute. CO2, a solute. Sodium, a solute. Potassium, a solute. You get it. If I'm not saying H2O, it's a solute. So water versus solids. Now let's go back to diffusion and talk about it first. Now, when we think about how important this is to understand, maybe it would make sense if we thought about why we want to know this, is that we have this liquid tissue called blood that is moving through the body, being pushed through the body. And most of our stationary cells, because most of our cells are stationary, they can't move, are going to be dependent on nutrients being brought to them and waste products being picked up and carried away and gotten rid of. So if you want to have a reason for knowing about these 2 terms, diffusion and osmosis, I hope that's good enough for you that you understand that all of our cells are dependent on having nutrients supplied and waste products picked up and carried away. So that's I hope a good reason. When you're looking at this blood vessel, this is a capillary blood vessel. I know it's a capillary because capillaries are 1 cell thick. That's all. Just 1 cell thick. And blood is moving through the lumen. This is the lumen, the opening of a vessel. Blood is moving through this lumen, and they're so small, the lumens are, that red cells that are being pushed through, red and white cells and platelets and nutrients and the plasma part, the extracellular part is called plasma. But cells are having to move through single file. That's how small this lumen is inside the capillaries. Now, Nutrients have to be able, and water, need to be able to move out across to our tissues rather quickly. And waste products would have to be picked back up rather quickly because blood is moving. It doesn't get to stop or slow down. It is moving. So let's talk about how that movement of nutrients into the tissue spaces and the movement of waste products back, how it actually happens. Diffusion, when we think about something called simple or passive diffusion, passive is usually a word that we often use when we think about, instead of simple, people will say passive. It requires no energy, none, no ATP. No charged batteries have to be used in this process, meaning you can save your charged batteries, your ATP molecules for other functions that the cell's carrying out, because this will not require any. Passive diffusion means that the solutes, if there's a lot of them in one area and not many in another area, they're going to just move down their concentration gradients from high areas to low areas, down the concentration gradient, just by like being at the top of a hill, letting go of a ball, and that ball's just going to roll down the hill. You didn't have to push it. It's just going to roll down the hill until it gets to a balance, right? No ATP, no charged battery needed for the process. So that is actually, again, known as passive diffusion, moving down a gradient. So if you have a lot of something on the outside of a cell and not so much of it on the inside of the cell, and you don't have a way of pumping it against the gradient, then it's going to move down the gradient. And it means that it's going to just move without any help. Now, we can also, our cells, and this is a huge difference between diffusion and osmosis, which I haven't talked about yet, but our cells are alive. And what our cells have the ability to do is to move things against the gradient, up the hill. Now, when that happens, when you need to do that, like as far as pumping sodium out, it means that you have to use ATP. You are going to have to expend energy. And your cells, your trillions of cells right now, every one of them are expending energy just in that way by pumping solutes against the gradient from high to low. A good example of this, and just to, so you know what this picture is showing, if this were an agar plate, and that's what this is supposed to represent, that looked like light blue right here, and the entire thing looked light blue. But if you took a little crystal, and this is what they don't show you, if you took a little crystal of dye that has a dye in it, like, I don't know, methylene blue or some sort of dye, and you took that crystal and shoved it into the agar, over time, what you would see the dye doing is moving out into the agar. It would just be called diffusion, passive diffusion. Now, is there any way that we could get that dye back into the crystal? Could we get it out of the agar and get it back into this crystal that's still sort of halfway there? It would take some energy, wouldn't it? We'd probably have to use some heat. We'd have to melt the agar. We'd have to filter it out, but we could get it. It would take energy though. So things moving down the gradient takes no energy. It's passive. But moving them against their gradient from low to high takes energy. I give the example in a classroom, if I were to spray perfume. If I were to spray a perfume, the people on the front row desk could smell it right away, because the solutes had moved through down their gradient. They're moving through the atmosphere, because there's everything, molecules all, atoms of elements and molecules all around us, even the air. And they would move down the gradient, and the people in the front row would smell it. Now, if I gave it another minute or two, probably the second row people are smelling it. I sprayed it, because it's still moving down the gradient. Took no energy. I didn't have to stand up in front of the classroom and wave anything. took no energy. It just moved. The solids inside the perfume, they're solids, move through the air. Now, then I usually ask the class, well, the people in the back row, can you all please get those solids, because there are probably a few back there already. Can you get all the solids and put them back in my bottle, please? And students will usually say, no, I can't do that. I can't do that. But they actually could. It would take a lot of work. I'd have to somehow set up an air vac that would pull the air from the room back through a filter, and then I'd have to have a filter, and I'd have to have something to extract it and purify it, and I could get it back in the bottle. But that would be moving it from low concentration to really higher concentration in the bottle, and that would take a lot of energy and work. Your cells are pumping things against the gradient molecules all the time. Is it taking a constant supply of ATP? Absolutely. And I've discussed aerobic respiration with you, and you would need to be able to know exactly what that is. That is the way our cells get a constant supply of ATP, by bringing in glucose, as a reminder, bringing in glucose, bringing in oxygen inside the cell. Remember the story in the arrow? You can make 36 molecules of ATP and give off CO2 and water in the process. But you have to have constant supply of ATP, and this is just one example of why. Because your cells are constantly moving solids up their gradient or against their gradient. So I hope that makes sense to you. Now, what about water? What about water, though? Okay, so understand that solids, as long as, and think about a red cell that has oxygen, load it with oxygen, but the tissues don't have very much. So oxygen just moves out. No energy required. CO2 would move back in. No energy required. That would be passive diffusion. That's just an example. But what about water? Water movement, we said, is not called diffusion. This is called osmosis. Now, it's a very, in one respect, the same as the movement of solids, but in another respect, and the most important one, very different. And you need to hear the difference too. Water does move down its gradient, just like solids did. So if there's more water in the plasma, which is the fluid part of blood, than there is in our tissue spaces, water just gently moves out, down its gradient. More water was here than here, so water moves out. Here's the difference between the movement of water and the movement of solids. I told you, and you understood, that your cells can move solids against their gradient, but your cells cannot move water against the gradient. That is an important distinction. Cells cannot move water up or against the gradient. So, osmosis is the movement of water between two areas of concentration, and water will move between those two areas until the water pressure is balanced between the two areas. And cells can't move water against their gradient, against the water gradient. They can't. So the reason that this is so important, this distinction, is that I need for you all to understand that if water is filling our cells, and water is bathing our cells, I should really call it fluid, bathing our cells, fluid filling our cells, and fluid inside of blood, even distributing nutrients and picking up waste from our cells, then the percentage of solids found, they're saying particles here, I'm saying solids. The percentage of solids found within these solutions, solution meaning fluid, had better be equal. It had better be equal, because if it's not, water will be moving. It will be moving from the areas that have the highest amount of water to the areas that have the lower amount of water, and there's nothing your cells can do about that. Now, for the most part, it all works well. It all works well until it doesn't. And how is that going to make a difference? Maybe you guys have heard the term edema. Edema, E-D-E-M-A. Edema is defined as an excess of fluid in the tissue spaces. An excess of fluid. Of course we know fluid is bathing our tissues, and of course we know our tissues are made of cells that are filled with fluid. Of course we know that, and we know water's kind of moving in and out and in and out. But how it moves is down its gradient. And if the osmolarity is off between one of those areas, the area that's bathing our cells versus the area of fluid inside of our cells versus the fluid in our blood, then water will be moving down its gradient, and it could lead to edema, which is an excess of fluid in the tissue spaces. And if there's more fluid in the tissue spaces than there should be, then it impedes how those tissues function. It can impede it enough that it ends up impeding how gases are exchanged and how nutrients and waste are exchanged, and the tissues will start to suffer. You all have probably heard, and you've seen people with edema, like in the extremities, like in their ankles or feet or hands, maybe in the extremities, and that can sometimes be due to gravity. But when you hear about pulmonary edema, that means there's an extra amount of fluid around the lungs. The lungs can't fill with air and can't expel, so that lungs can't move. If they can't move, they can't move air. You've heard about cerebral edema. That means there's an excess of fluid around the brain, the central nervous system. Pulmonary cerebral edema, not good situations ever. There can be an excess of fluid around the heart. This again is not a good situation. So we need the osmolarity. We need the osmolarity between these compartments to be balanced, because we know that fluid, that water moves down its gradient, and there's nothing your cells can do about it. So what I want you to know now are a couple of other terms. I want you to know the term tonicity. Tonicity means the ability of a solution, a liquid, the ability of a solution to affect the volume and pressure in a cell. So to affect, because we know cells are filled with fluid. So tonicity is simply meaning the ability of a solution to affect cell size. That's what tonicity means. Now, what I want you to look at here is this middle picture. This middle picture is a picture of a normal red blood cell. Red blood cells are discoid in shape, meaning they're shaped like a disc. They're really thin in the middle. And so sometimes it looks like a little hollow center, but that's the way they're supposed to look. They're very flexible, red cells are. They're squishy. They can go through capillaries, single file. This is a normal red blood cell in normal solution. Actually, the tonicity of the solution is equal, iso as a prefix means equal to the inside of the cell. So water is actually moving kind of gently in and out of the membrane, and it's not causing the cell size to change. It's not putting pressure on the cell. This is perfect. Okay, so isotonic solution. What would happen if I took this normal red blood cell that should look like this middle one, and I put it over into a hypotonic solution? Hypo as a prefix means low, and in this situation, when you're talking about tonicity, it means low solutes. So in this solution, if there's not many solutes, it means there's a lot of water. The percentage of water is higher than the percentage of solids. Hypo solid. Hypotonic means low solids. So if you took this normal red blood cell and dropped it into hypotonic solution, the water from the solution will move into the cell and it will blow it up. It will blow it up until it is really no longer the shape that it should be. The shape structure dictates the function. So this red cell is not functioning the way that it should. Red cells deliver oxygen to our tissues, which we desperately need for ATP all the time. If you don't believe it, cover up your nose and mouth. You'll see how long you last, not long. So this red cell's not functioning in a hypertonic solution because it is swollen up. It could actually get so swollen that it ruptures and lyses. Now, what would happen if I took this normal red blood cell and put it into a hypertonic solution? A hypertonic solution has a lot of solutes and not much water. So the water that's inside the red cell leaves. The water, because water goes down its gradient, there's nothing that our cells can do about it, even though they're alive. They can't do anything about water moving, osmosis. This is the way it happens. In this situation, the red blood cell has shrunken. It's crenated. It's shrunken up. Again, do you think this red cell can function? No. No. So crenation means shriveling, shrunken, and that's what happens to cells that are in hypertonic solutions. So when people need fluids, it's very important that, and they're getting fluids, it's very important that you're thinking about these types of things. Like, what is the osmolarity of these fluids? Is it isotonic? Is it normal saline? Is it an isotonic type of solution? And you have to be careful in how fluids are actually given to patients, because the osmolarity of a solution can affect the cell size. Okay, so I told you that active transport means that you're, maybe I didn't, but this is what it's called. Active transport is when your cells have, and they do, have the ability to move solutes, particles, against the concentration gradient. So from low to high. And that can happen. But what does it take? It takes energy in the form of an ATP molecule. So energy is going to have to be consumed for that to happen. One of the ones that we, again, example that we always give is that sodium potassium pump that all your cells are using all the time. Those pumps are working 24/7 in your trillions of cells, sodium, potassium. But there are other pumps as well. There's potassium, there's chloride we talked about, and we do understand that those other pumps happen as well, but the sodium potassium is the major pumps that we think about. So when we think about our body being able to keep, body temperature, even when the outside temperature is a little cooler, can't be too cold, but a little cooler, we know that it's a lot to do with how our cells are alive and are functioning. These pumps are working, and every time any sort of chemical reaction is occurring, heat's given off. So anyway, all right, so all of this is necessary for anything that's requiring energy inside of our cell, not just nerve and muscle function, but certainly that's important too. There's a term on this slide. That's the only reason I included these slides. There's a term here that I want you to get at this particular part, and it's called phagocytosis. Phagocytosis is the process of a cell eating. The ability for the cells to actually, and here's a cell, to take in a particle, and it attaches to special receptors on the outer membrane of the cell. It can actually ingest it by surrounding it with the membrane. And again, see how we change our membranes out all the time? We have specialized enzymes, digestive enzymes, found within particular little vesicles called lysosomes that I haven't talked about, but I will in a second, that can chew it up, digest it, and then literally spit out the residue. So phagocytosis is the process really of a cell eating. And many of our cells have the ability to use this process for different functions. Some of them are immune system cells that can phagocytize to help get rid of debris and foreign particles. But this is how it does that. Now, if we were to look at it on an illustration, we can see it here. But look at the electron micrograph of your actual, of a little tiny piece of a cell. So this cell would be huge here. A little tiny piece where we're just looking at the membrane, the outside particles, the intracellular, extracellular, and you can see how these particles have actually attached to receptors, and now it's the whole membrane is folding in. And now what had been outside of the cell is going to be able to be used inside of the cell. This is actually referred to as endocytosis, the process of a cell bringing something inside. We actually can do the opposite of that as well, and I think that is, exocytosis is here, is a slide here, where our cells can make products and then actually attach those to the outer membrane and release them through secretion. So you can actually see a cell doing that here, releasing its product. All right, now the inside of cells. What's on the inside of our cells? We've just talked a lot about what a plasma membrane is made of, the organic structures, the lipids, predominantly lipids, the proteins, the carbohydrates, the specialized structures, the ability of the membrane to be permeable to solutes, diffusion happening, and water moving, osmosis. So we talked a lot about that outside of a cell. We talked about structures that some cells have, microvilli, others have cilia, sperm have flagellum. Right, so we talked about those. But now let's go inside of cells. I'm going to take a quick break and I will be right back. Okay, I am back and we are going into the cell, inside of the cell, and we're now going to start looking at the structures that have very specialized functions within the cell and taking a look at those. The cytoplasm, we said, is going to be that fluid matrix, the intracellular matrix that is keeping the integrity of the cell, the osmolarity of the cell where it needs to be. And this is where we're going to find organelles, the cytoskeleton, and even some inclusions and vesicles. They're in this fluid that is sometimes referred to as cytosol. So when we look at an illustration, in this particular illustration, the apical surface has microvilli, so you can look at these a little bit closer. But we're going to go inside and take a look at some of these structures. I am going to have you all do a lab where you're drawing. You have a word bank of with some of these structures, and you will be drawing and coloring and labeling by hand these structures. It's understood that people who use these techniques, the kinetic energy of drawing, coloring, and labeling, it can help them to actually learn structures from doing that. So that will be a lab component of this, but those instructions will be in that lab. But this is us looking at them right now together in this lecture setting, so, and hopefully me explaining to you how they function so that you have a good working knowledge of these structures and also their functions. So we're going to take a look inside. When we think about the cytoskeleton, if you look there, looks like there's this, I call it the highway. Some people refer to this as the scaffolding of the cell, but it looks like all of these little threads, but they truly are like highways between these different structures. And it is how things move within the cell, so it truly is like a highway. If we looked at an electron micrograph of these cells, these are actually stellate cells, sort of star-like in their structure. But if you look, you can see it almost looks like little hairy woven areas inside. But again, these are the cytoskeleton. So when you think about one of these structures being blown up for you to see, it would look like this. And I do want you to know that this is giving structure to the cell, integrity to the cell, but mostly how substances are moving, solutes are moving between particles between one area and another area. The products that are being produced in the cell, because cells are essentially factories, how they are moving from one area to another. So it is with the cytoskeleton. Let's take a look at organelles now. When you think about organelles, we know that there are membranous ones that actually have membrane-bound structures, and then there are non-membranous organelles as well that we'll look at. So we'll take them one at a time. When we think about the nucleus of a cell, and depending on the life cycle of a cell and the age of the cell and what the cell is differentiated into, nucleuses can look different in one type of human cell from another type. But as far as their function, what I need you to know as far as their function is that the nucleus of a cell houses, stores, organizes the DNA of a cell, the cellular DNA. It's important to put cellular DNA. So the DNA that you inherited, half from your mother, half from your father, that holds all the recipes for all your many different types of cells and how they're going to function, that cellular DNA is housed in the nucleus. And the nucleus is a membrane-bound organelle. So that is what I'm going to want you to know about it. Now, when you think about the DNA, you usually hear about DNA as chromosomes, don't you? Chromosomes are DNA, cellular DNA, but when they're actually cellular DNA in duplicated form, and a lot of people don't know that, but when the DNA is not in duplicated form, when it is just spread out and is functioning for the cell, it's called chromatin. So it's in chromatin form. When the cell has to divide, the DNA has to make a copy of itself and condense down, then it's in chromosome form. So if you ever hear the term chromatin or chromosome, then you know you're really just talking about the cellular DNA. Nucleoli, and if we go look at the nucleoli, this little structure here that they've made pretty obvious for you. If you go back here, it's this one. Nucleoli, or nucleolus singular, are going to be that where ribosomes are made. Ribosomes, and we'll talk more about ribosomes in just a second. But this is where ribosomes are made, is in the nucleus, in a very special site of the nucleus called the nucleolus, or nuclei for plural. If we looked at a electron micrograph and looked at this nucleus, we can see the entire nucleus is here and the nucleolus is here. Nucleoplasm just means that matrix, that's that fluid matrix that the chromatin is in. And if you look at this grainy looking structure here, that's just the DNA that has been sliced and stained, and we can see it on an electron micrograph. If we hadn't cut into the nucleus here, we were seeing the outside of the nucleus. It's very porous looking, and that's because the instructions that need to be carried out in the cytoplasm, the instructions for the cell, have to be able to come through those little pores. All the instructions for all of your traits are found in the DNA chromatin of a cell. But the recipes, the instructions have to come out one at a time. They can't get out of the nucleus. So where they come out through these little pores. So again, this nucleus that you see, it's green here, is very porous, so that the instructions that are being sent out from the nucleus to be carried out in the cytoplasm can move through those little pores. The entire recipe book can't, but one instruction at a time can. So I hope that makes sense. Now, So when the instructions, one recipe at a time, comes out of the pore, where does the instruction go? It goes to a organelle known as the endoplasmic reticulum. The endoplasmic reticulum, and it's been cut here, a wedge of it's been cut here for you. There's two parts of the endoplasmic reticulum. The part that is actually really close to, kind of continuous with the nuclear envelope, the nuclear membrane, is called rough. It's called the rough ER. The reason it's called the rough ER, it's kind of hard to see on that picture. I'm seeing if I have a better picture. I don't. The reason it's called the rough ER is because it's embedded with ribosomes. So this membrane has been studded with ribosomes. And I had just talked about ribosomes being produced in the nucleus, and then they leave through those pores and embed in the rough endoplasmic reticulum membrane. The ribosomes, the function of a ribosome is to act as the workbench, literally a table workbench molecule of where proteins will be built. Wow. So ribosomes are embedded in this endoplasmic reticulum, the area called the rough. And those ribosomes are the actual tables, the workbenches, where proteins are going to be built. Now there's also a smooth, these are the little ribosomes embedded. That's a picture I wanted. Now there's also a smooth endoplasmic reticulum. So the rough was here. You can see the rough kind of looking here. And then the smooth out here. The smooth are going to be where lipids, fats, are made. You can kind of think about fats as being kind of slippery. And so there are no ribosomes here. The ribosomes are where proteins are going to be built. And remember, proteins are made of monomers called amino acids. So amino acids will be brought to these little tables, these little workbenches, and as the chains start to form, you know, we're getting a polypeptide chain of amino acids, which is our beginning of our proteins. Those workbenches are called ribosomes. The smooth ER lipids are made. Wow, that's really nice. So now we have the rough ER making proteins, the smooth ER making lipids, and then where are those products going? Where are they going to be sent to? Remember, it's a factory. Where are they going to be sent? The recipes came from the nucleus. The recipes did, one at a time, to the workbenches, the ribosomes. But where are the products going to be sent? Are they going to stay right here and build up and build up and build up? No, because if you look through, we've got all these highways, the cytoskeleton. They're going to actually be sent to what is called the Golgi Complex. The Golgi, some people say Golgi. The Golgi Complex, some people say Golgi body, Golgi organelle, or just Golgi or Golgi, either one, whatever. They are sent to this organelle for wrapping and shipping. Wrapping and shipping. So again, the nucleus sends out a recipe. It goes to a workbench called a ribosome. Protein, if it's a protein being made will be sent to the Golgi for wrapping and shipping and being sent out of the cell. Sometimes they're going to be sent just throughout the cell to be used within the cell. Sometimes they're going to have to be sent out of the cell. But what wraps and ships them? The Golgi does. If it's a lipid, being in the smooth endoplasmic reticulum, same thing. Same thing. It will be wrapped and shipped by this Golgi complex. It's wrapping and then shipping in the form of these vesicles, these little vesicles that are surrounded by a membrane. I want to go back and show you free ribosomes, because ribosomes are not only going to be found embedded ribosomes are not only just embedded in the rough ER, sometimes ribosomes are actually also throughout. You see they look kind of green right here, these little tiny workbenches. So proteins are actually being made out here on these free ribosomes as well. Our cells are essentially protein factories. They really are. Now, so thank you, Golgi. This is an electron micrograph of one, but here's an illustration of it. And after a product arrives to the Golgi, it gets wrapped, pinched off, shipped by way of the cytoskeleton to wherever it needs to go. I want to tell you about a couple of specific types of vesicles that we know most cells have, all cells have actually. These vesicles, and these are in addition to whatever the cell has differentiated and matured to become, but these are in addition. The first one vesicles are lysosomes. Lysosomes are actually going to contain, these vesicles are going to contain digestive enzymes. These digestive enzymes are going to be able to break down products for recycling. So they're going to be able to break down products for recycling and even sometimes getting rid of foreign materials. So this is a very important function of lysosomes. Another important function is going to be that they are part of cell death. You have, I told you there's some of your cells are only supposed to live for a few hours. Some of them days, some of them, longer. But part of the cell dying is important. If not, if you lose that ability, if a cell has lost the ability to self-destruct and is just stealing nutrients from the environment, has lost its cell parameters as far as how it's going to grow, we call that cancer, don't we? But most cells do what they're supposed to do, and when instructions come for the cell to self-destruct, it actually does. So lysosomes play a role in that too. So they very much are needed in the cells. They must be functioning to help clean up, clear up, recycle old, worn out products, but also program cell death. There is a disorder of the lysosomes that is inherited. It is a faulty information for lysosomes of the nervous system cells. It's called Tay Sachs. I think you have a deeper insight in this chapter. It's not this chapter, it'll be soon. But it's a disorder of that's inherited that is 100% fatal. And children that are born with Tay Sachs inherited disorder are going to look like they're developing okay in early toddlerhood. They're meeting their milestones. But then all of a sudden, the central nervous system can't get rid of byproducts that are building up. They can't clear them and recycle them. So the central nervous system is what's affected first, and it is a neurological, progressive, terminal disease. These children usually don't make it to 5 years old. It's kind of a horrific death. And as I said, 100% fatal. Where's it happening? It's happening at all disease happens at a molecular and cellular level. Where is the problem? The problem is in the recipes from these lysosomes not functioning the way that they should. It's a devastating disease. So there's another type of vesicle that our cells produce that actually are going to contain peroxisomes. These are called peroxisomes. Now what these do are actually contain antioxidants. And as we learned in chemistry, antioxidants help to neutralize free radicals. So these are again incredibly protective for our cells. And they're something that we produce naturally because free radicals are happening just in normal metabolic activity. Certainly we create more free radicals when we do things like take in substances that we know to be drugs. Alcohol is a drug, nicotine, all kinds of other things like food preservatives and colorings and dyes. So those types of things. And we do know that that peroxisomes are very protective for us in neutralizing free radicals. And they really wouldn't look any different from another type of vesicle, but they're just showing you how your cell uses them and sends them throughout the cell to use to help to neutralize free radicals. Again, here is just examples of a cell looking inside of a cell and seeing what some of these vesicles actually look like. That's sounding like a lot of work our cells are doing. They're doing tremendous amounts of work all the time, 24/7. And to do work, we have to have very specialized organelles called mitochondria. These mitochondria are where we get the majority of the ATP from cellular respiration. The second step and the third step of cellular respiration, the first step was glycolysis that's happening in the cytoplasm. But the second and third step happens in the mitochondria, and this is where we get 34 of the total 36 ATP recharged batteries. So the mitochondria are said to be the powerhouses of the cell, because they essentially are the powerhouses of the cell. And it's very important that these actually be functioning. Now, a unique thing about mitochondria in our cells is that we actually get the mitochondria themselves, these organelles themselves, have their own mitochondrial DNA. They have their own mitochondrial DNA. You can see it here. Now, that means that they kind of have their own set of instructions inside of our cells. Remember, the cellular DNA that you got from your mom and dad that tells the cell what it's going to become, how it's going to produce the products that it needs to produce. Everything about it is coming from the cellular DNA, but the mitochondrial DNA is different. And it's in those mitochondria. The mitochondria have kind of their own set of instructions. Where do you get your mitochondria from? Where do you get it from? You get it from your mother. It is the egg that has all of the cytoplasmic inclusions and organelles that the zygote is going to use to survive. The egg has half the number of cellular chromosomes that came from the mother and is waiting for the other half from the sperm to be delivered from the father to make up the nucleus DNA. And without that, the egg dies within a few hours. Without that sperm contributing half of the cellular DNA, the cell dies within a few hours. The egg dies. But if the sperm gets there, then the cell itself has what it needs. The zygote has what it needs, including the mother's mitochondria, that the process of mitosis and life can continue. Okay, first, the cell life. Okay, so it is important to understand that, because we know that mitochondria has its own mitochondrial DNA. This actually contributes to the information. It's not the only thing, but it contributes to the information that has helped us to understand how complex life, multicellular, complex life, evolved on this planet. We know that life has been on this planet for at least 3.8 billion years. 3.8 billion years at least. The evidence shows that. The earliest life, though, were single-celled, simple life, similar to bacteria that have no organelles, no complexity compared to what our cells have. So how did life go from simple singular cell life to organisms that had the ability to have the energy and the complexity of cells like our cells and plant cells and fungal cells, fungi? How? This explanation is in the, it's actually called the theory of endosymbiosis. Endosymbiosis is one word. This is a scientific theory, so it's, an exhaustive explanation of how complex multicellular life evolved from simple single-cellular life. And one of the things that support this explanation is that we have these organelles like mitochondria that are not alive. They're not alive, not on their own, but they have their own DNA. There's another organelle that's found in plants that also has its own DNA. That organelle are chloroplast, where photosynthesis occurs. So once upon a time, sounds like a fairy tale, but once upon a time, these organelles probably were alive and were independent, the mitochondria and the chloroplast. But now they have been taken on by these complex cells, and now they're dependent on them. But our complex cells, are also dependent on them. So endosymbiosis, symbiotic means living together. And this created the ability of cells to produce massive amounts of energy to do more complex work. So again, mostly what I want you to know is that mitochondrial DNA contributes to the theory of endosymbiosis. And your mitochondria came from your mother. Your mitochondria that came from your mother, if you have any siblings that were from your mother, you all have the identical mitochondrial DNA. Mitochondrial DNA is actually being used a lot now in forensic science. Forensic science, because it's easy to do lineages through mitochondrial DNA of relations and kinship. So it is absolutely easy for that way. There are some inherited disorders of the mitochondrial DNA. So that they can be kind of horrific, actually. So they are pretty rare, though. Now, thank you, mitochondria. Thank you for the powerhouse. Thank you for supplying the energy for our complex cells that are absolutely carrying out so many different functions that we call being alive. So that's what's giving us that. The centrioles are these structures that they look like little, I don't know how you would call those. There's candy that looks like that, but okay, maybe I'm hungry. These centrioles have a major function of being necessary when a cell divides. So what I want you to know about centrioles, and we'll see them again in the next chapter, and when we're talking about genetics and cell division, but these centrioles aid in cell division. So when this cell is going to need to divide, these centrioles are going to play a major role in that, and we will see that. Now there's tons of inclusions that look like little vesicles on just an electron micrograph, but the inclusions can hold things like glycogen, which is a storage form of glucose, can hold pigments, fat, and sometimes can even hold foreign bodies as well. So there are all kinds of inclusions that you can look at. I really only talk to you about like lysosomes and peroxisomes. Now, of course, though, we do know that all those other structures that we talked about would be important to note. When I go back and look at a cell and look at a wedge of a cell, and we actually go all the way back and look at how much is labeled in a cell. On first glance, this may have seemed intimidating, but I hope that you all will not think it's intimidating really not intimidating now that we have gone over all of it. If I were to tell you that, I'm going to get you to draw a cell and color and whatnot, I am not judging this on artistic ability. And just to prove that to you, I'll show you my artistic ability, which is kind of lacking. But truly, I want you to be thinking about a cell as a factory, because they really are. So if you were to draw a cell, it wouldn't matter what structure you drew, some are round, some are square looking, some are whatever, but you would label the plasma and you would write it out, the plasma membrane, so you know you would write that out. Then obviously a cell is going to have a nucleus, isn't it? And you would label, write out the word nucleus. You would maybe even include the nucleolus, maybe. And then you would draw the structure of, look at me, I'm not drawing it very well, but the endoplasmic reticulum, because we said the nucleus houses the DNA. We know that it does, but we know that on this rough endoplasmic reticulum, that's rough because it includes these ribosomes, workbenches. Some of them are even free out here, aren't they, in the cytoplasm. And by the way, you would have labeled cytoplasm, wouldn't you? You're going to know that, and you would label the ribosome. You would label this as a ribosome, right? Then you would say this is the smooth ER, and you'll be able to write better than I can. You would say that this is the rough ER, because it has the ribosomes, the workbenches, right? You would even label, would certainly label the Golgi apparatus. I think you would be able to draw this much better than I could. But you also want to use colors, because colors help us to remember. And when we're doing something, if we're using colors, it's another tool for memory. You would actually want to also include certainly your mitochondria. I could draw better without a mouse, but anyway, the mitochondria, you would label that, write that out. And you know, you would draw the centrioles, you'll draw anything that's on the word bank. Everything on the word bank would have to be included in your drawing. And then once you draw them, if I were you, because these are your drawings, I would jot down like what they do. So like the nucleus, I would say houses the DNA, cellular DNA, cytoplasm, the fluid matrix of the cell. I would say ribosome, the workbench for where proteins are made. The rough ER, proteins are made. The smooth ER, where lipids are made. Mitochondria, energy. Golgi wraps and ships the products in the cell. You might have to, on your cell, also put like the mitochondria, so you might have a little area, excuse me, not mitochondria, the microvilli. You might also on the outside of your cell include these long hair-like structures and label those the cilia. You know, you might even put a really long tail and say flagellum. So there's no real cell that would ever look like this, but that's how you would do it. And again, if you jotted down even a brief explanation of what they do, that's just another tool for you to try to get this information. So with that in mind, I'm going to go ahead and close out our lecture on cytology, and I really appreciate y'all's attention. If you know, reading the textbook is great. I think sometimes the textbook definitions and terminology can be a little confusing. I hope I've broken this down for you, though, that you have a really good idea and working knowledge of how our very complex eukaryotic cells function in our bodies.